on the chat by Tainá and Yesid about the third call for Cabana secondments is now open to 21 January 2021. I don't know if this is for the next year, but that they put a link on the chat to let us know about this online projects and exchanging experiences. That's very interesting. Thank you, people. Shall we start, Miguel?
Yeah, let's go.
Okay, great. Then it's a great pleasure to have Irene here again with us. Just share my screen. Well, I'm not able to share my screen, Miguel, to introduce Irene. Well, still not able. I don't know if Miguel has to let me share my screen. Who is organizing? Miguel. Well, where is Miguel? Tiago, você está aí?
Oi, professora, pode compartilhar.
Ah, tá, obrigada. Yeah. Obrigada. Sorry. Well, then it's a pleasure to have, yes, sorry. A little bit, well. Well, I'm not seeing anything. It's a black box. Let me try again. Sorry, technical problems. Well, is it working now?
Yes.
Yes. Okay, sorry. Sorry. Then it's a pleasure to have Dr. Irene Papatheodorouker from EMBL-EBI in United Kingdom. Dr. Irene is a team leader, and his specialty is gene expression. She leads the gene expression team, which focuses on gene expression analysis at tissues and single cell levels across species. Her team delivers tools and service for the submission, achieving analysis and visualization of functional genomic experiments. After completing her degree in genetics at University College London, Irene focused on bioinformatics through the master and PhD students at Imperial College. And then it's a pleasure to have a bioinformatician with this experience in our seminar, the BRAZWEBINARs, and will be very exciting for us to know about the single cell data bank and everything that Irene is going to tell us today. Thank you, Irene, for being here. And I hope everything works very well now.
Thank you, Gloria, for the introduction. And thank you, everyone, for the opportunity to speak here again. And I'm going to share my screen now, my personal. So can you please confirm that you can see the right version of this, the right side of the screen?
Yes, it's okay, Irene.
Excellent. Okay, so thanks very much. And thank you for the opportunity to talk about the work in my team. So as Gloria said, I'm a team leader at the EBI, and my team delivers Expression Atlas and Single Cell Expression Atlas. And also I lead a small research group that complements the activities of the Expression Atlas team and utilizes the data that we collect. So just to start and give an outline of this talk, I'm basically going to talk a little bit about the services that we develop in the team, Expression Atlas for single-cell RNA-Seq especially, which is the latest addition into our services. So I'm going to go into quite a detail there. And then I will talk about a project of an ongoing unpublished work of a postdoc of mine, which utilizes publicly available expression data sets to map late life associated comorbidity genes and processes. And then I'll touch on a bit on future projects and finish the talk. So overall, the mission of my team is to extract biological meaning from gene and protein expression data and make it easily available to the research community. For this purpose, we developed the Expression Atlas. This is an open science resource for querying and visualizing gene and protein expression across different species, tissues, and conditions. And we do that by collecting expression data from the public domain, from archives such as GEO, ENA, and ArrayExpress or BioStudies. And we perform extensive data curation. So we look at the metadata that accompany the data sets and determine whether these are complete, whether we can reanalyze them and reanalyze the data based on this metadata. And then we perform standardized analysis on the raw sequences and the RNA-Seq. And we select the high quality data for this. And then the high quality analysis results, we index them and we map them with different ontology identifiers and make them available through the user interface of Expression Atlas to enable search across the different data sets and also visualization of the results. So we don't discriminate a lot on the sources of data, so that go into Expression Atlas only on quality. So they come from many different species and tissues and different conditions, whether this is treatments or environmental stresses and so on. And the idea is that through this resource, any user of any background can discover and interpret gene expression analysis results quickly and easily. So the Expression Atlas, which is the original database, it looks like this, and you can access it from the URL at the bottom. Currently, in the last release of Expression Atlas, we have data sets from 65 different species, and that is over 4,000 bulk data sets, of which about 227 are of baseline expression. By baseline expression, I mean the constitutive expression levels of genes in tissues at normal conditions. And also we include about 3,800 differential expression data sets where two different conditions are being compared. So if you query, you can query Expression Atlas for a gene, for example, or a condition. Here I show an example of querying the gene reg1b. And what you get is really a list of heat maps on the different species where this gene is expressed. And here I have selected the human one. You see a little anatomogram for some of the species on the left-hand side, and on the right-hand side is a heat map that shows the different tissues on the on the columns, and the lines are the different baseline experiments where this gene appears to be expressed. What you can see from you can probably recognize GTEx here and ENCODE amongst the other experiments, and also apart from transcriptomics, we do include some proteomic experiments as well. So what you can see here is that in all the experiments where pancreas is included, this gene is highly expressed as opposed to the other tissues. And that also follows on the proteomic experiments as well. Now, there's a second tab, the differential expression, where you can click and find all the different comparisons where reg1b is upregulated or downregulated, and they're ranked by the log 2-fold change. And there we, by inspecting this list, one can quickly see that a pattern emerging that reg1b gene appears to be downregulated in pancreatic cancer, amongst other things. So, so that was the bulk side of Expression Atlas. So the last few years, with the emerging single-cell RNA-Seq dataset, we transformed this resource and included a section that caters in similar ways for single-cell RNA-Seq dataset. And this is what I'm going to go into more detail now. But first, taking a step back, it's important to motivate choices. Important to look at gene expression in single cells. The cell is a basic unit of life, with cell types differing substantially across each tissue. With bulk RNA-Seq, which is shown in this lower part of the diagram, we can only achieve the average expression from all the cells as they are lumped together. Whereas with single cell RNA-Seq, we have the ability to dissociate the different cells and take readouts of each one at the time, and therefore reveal the heterogeneity and subpopulation expression variability of thousands of cells at the time. Obviously, this technique opens the door to several biological and clinical questions. So we can understand, it gives us the tools to understand heterogeneous samples, such as analyze cellular heterogeneity during immune or stem cell development. And also it enables us to identify and analyze rare cell types, such as circulating tumor cells from liquid biopsies, understand cellular transitions and switches in cell state, and also work on comprehensive genetic diagnostics. Single-cell RNA-Seq has already revolutionized many fields in biology in recent years. It's increasing the resolution of our understanding in areas such as neurobiology, microbiology, embryology, and so on, to just mention a few. So now I'm going to go on to how we developed this integrated resource of uniformly analyzed single-cell RNA-Seq data, that is the Single-Cell Expression Atlas. So we already had established a nice workflow for getting, curating, analyzing, and visualizing bulk RNA-Seq data. So we followed the similar workflow, and we reestablished our workflows for the data curation for single cell data. We developed new pipelines for analyzing the data. And we also ran extensive benchmarking to identify pipelines that and sets of tools that work that work well across the different samples coming from different species and tissues and conditions. And then we also developed a user interface for the visualization of different cells and single cell data that is quite different to what a user would need to visualize from bulk. So on top of that, so we developed the single cell expression atlas a couple of years ago, and now we're using it to also power up and disseminate different large-scale projects, such as the human cell atlas and soon the fly cell atlas as well. And just to go ahead, and this is the front page of the single cell expression atlas, and you can find more information about its development through our latest nucleic acid research update, the latest database issue. Currently, the single cell expression atlas has information from 18 different species, and that includes 217 single cell datasets, and that consists of over 5 million cells. Most of the datasets are from human and mouse, and that's really a reflection of the majority of the datasets that exist currently in the public domain. Then I did mention that we had to work a lot on developing the necessary metadata that that any experimentalist wishing to publish their single cell RNA-Seq results need to disseminate with the raw data to enable this standardized reanalysis, any kind of reanalysis really. So, so we compiled a set of guidelines together with our colleagues in the Human Cell Atlas for reporting these single cell RNA-Seq experiments. And these guidelines are used for any single cell submissions to RA-Express and ENA Biostudies. And the Human Cell Atlas is using them as well. And they describe the sample, the single cell isolation, single cell well quality, the library construction, whether they have barcodes, UMIs, and so on. And also it includes some post-analysis, single cell quality, and the inferred cell type, which is basically the annotations of the cells by the data generator by the experimentalist. So as we collect this metadata and we reanalyze the data, then we developed a user interface to visualize these experiments. And that's here I'm presenting the results of this similar query. Here I used the reg1a gene. And I dug into a pancreas experiment where one can visualize this experiment with two versions of a t-SNE plot. On the left-hand side, the cells are colored according to the metadata, and in this case, it's the cell type. And on the right-hand side, the cells are colored according to the expression level of reg1a gene. And what a user can clearly see here is that the reg1 REG1A gene is highly expressed really in a particular cell type, which is the pancreatic acinar cells. Then we also provide different views of the data, not just the t-SNE plots, but you can click on the anatomogram here. For some tissues, we have images available of the organ, and these are zoomable images for specific regions where the experiment was sampled from. And on the right-hand side, you can visualize the different cell types and their marker genes as they appear in the experiment. So, for example, if we click on this little area here and zoom in, we can look at these pancreatic acinar cells that get highlighted when you click on them, and then REG1A is there, one of the marker genes for that cell type. So in collaboration with our colleagues in the Human Cell Atlas, we have developed this kind of anatomograms for various other tissues, such as the placenta, liver, lung, kidney, just to name a few, and we continue to develop more. So in the future months and year, there is going to be more of these anatomograms available for easy exploration of the results of the Human Cell Atlas and other human data as well. So now, apart from recording metadata correctly and notating them with ontologies, and also developing the user interfaces to easily interact with the data, in order to generate these nice integrated views, we also need to perform a standardized analysis. And now I will just spend a couple of minutes to describe a little bit the analysis pipelines that we use. So single cell expression atlas takes in data sets from plate-based studies, such as the SMART-like protocols, and also from droplet-based studies, such as 10x or DropSeq. And that means that we need to have two different analysis pipelines for generating the counts table. So our pipeline for plate-based studies involves the use of Callisto. So we do read filtering and trimming, read pairing. We quantify the data using Callisto. We remove empty droplet, well, that's for the droplet studies, and aggregate our libraries and samples into a row counts table with all the cells. For droplet-based studies, we use a tool out of the box called Alevin to perform all those steps, and then we use droplet utils. And we also generate a similar row counts table with all the cells present. And then for what we call a tertiary analysis, which is the cell filtering, assessing the quality of the cells, performing normalization of the data in the counts table, performing dimensionality reduction, clustering the cells, identifying markers in the different cell types, and also performing batch correction. We employ the tool, community developed tool called ScanPy, and also Harmony for the last step of batch correction. And so our pipelines are based on community developed tools that are maintained well by their developers. And we have also wrapped them and made them available through Galaxy in collaboration with our Human Cell Atlas colleagues. So if you wish to run your data set, for example, analyze it using the single cell expression atlas analysis workflow, you could go into the Human Cell Atlas, use galaxy.eu, and make an account, upload your data, and use the workflow that we provide here that is the same as used by the single cell expression atlas. Or you can use the Human Cell Atlas workflow, or you could develop your own workflow by using the different tools and stringing them together in Galaxy. So you could read about more about these tools and workflows from our paper of this year. And obviously we would be very happy to help in the team. So, last, as I briefly mentioned before, we try and aggregate and include the data sets that are valuable to the wider research community, and that means we also disseminate the results of large-scale projects such as the Human Cell Atlas, soon the Fly Cell Atlas, and Tabula Muris as well, which is the mouse atlas. Now I would like to switch a little bit and talk about how my team also uses these data to perform add value to them further and try and answer some more biological questions. So basically, thanks to all the work we're doing in Expression Atlas, we are collecting and performing standardized analysis to thousands of expression datasets for a resource, and my research team uses data to take it further and also investigate the gene expression to phenotype connection in human disease and also across species. So we have a couple of different research themes in the group. One is exploring disease phenotypes. So we try to map pathogenic cell types, disease types, using techniques such as cell type deconvolution. We also work with colleagues to try and identify these aging-associated comorbidity effects, and also work with colleagues on the Crohn's disease cell atlas as well. And on the other hand, as we collect all these different data sets, the cell atlases from the different species, we're starting to compare these, so the cell types between fly and mouse and human, homologous tissues, for example, in order to understand the similarity and difference between model organisms and humans, with the future plan to develop a kind of cell type expression orthology that could perhaps help researchers relate experiments more accurately from model species to human disease and so on. So just to talk a little bit about spent a very little bit of time on an ongoing project in the team, which is to map pathogenic cell types to disease phenotypes. The idea here is to leverage a large amount of bulk expression studies on hundreds of diseases that we include in the bulk expression atlas and infer their cell type proportions using cell type deconvolution techniques. So there are many deconvolution techniques available, some rather old that were used in the days of the microarrays, actually. They use, the majority use some version of support vector regression or version to assign cell type proportions based on marker genes per cell type. And now with the single cell, there's a lot of room for improvement. Also, we see a boom in new methods being developed. So what we are planning to do here, well, this is actually underway. We're doing on the available techniques to try and identify tools and a set of marker genes that are suitable to identify cell type composition profiles of disease samples in tissues from expression atlas. And also would like to take this further to look into disease relevant processes at cell type resolution, using a network approach, and then combine this with EQTLs. So, and then now I would like to focus the last part of the talk in projects on the project of my postdoc.
Miguel.
Eu acho que caiu
we
lá na
lost her
Grécia.
again.
Yeah, we lost Irene in Greece.
She's in Greece now.
She's in Greece now.
God.
The communication was going bad.
Bad. Yeah.
Hello.
Oh, hello.
Hello. How much did you miss?
No, we just missed the last final part. Yeah.
Okay.
You are about to start talking about the postdoc project.
The postdoc project with Janet.
All right. Great. I will reshare. I switched off my camera for this reason. So let's continue.
Great.
All right. So this is basically unpublished and ongoing work of Madoulikas. So here we are motivated by the fact.
Irene, Irene, we can't see. Yeah, now we can see your screen. Okay, go ahead. Now, now we can see it. Okay, go ahead.
You are experiencing some delay in the screen, but now we see Madhulika Misra and the title, not the graphs.
Excellent.
Okay.
Perfect. So here we are in this, and now you can see a graph, right?
Not yet.
Still waiting, yeah.
Yes, now we can see the graph.
On the left.
Perfect. I see there's a little bit of a delay. Yeah. So okay. So here we are in this project. We're motivated by the fact that the occurrence of multimorbidities, which is basically what happens in individuals with increased age. They tend to have more than two disorders, conditions at the same time. So this increases substantially with age, and as a result, the number of medicines one has to take also increases with age, causing in some cases significant side effects as the diseases have been studied individually. So what we are keen on identifying is what is going on at the molecular level. So that has been studied before, and here is an example from the literature, from the Human Diseasome paper, where the authors have explained what happens at the molecular level in case of multimorbidity. So here you see basically a panel of different diseases and how these intersect in the different genes that are associated with these diseases. And then you can find some disease pairs that are actually related anatomically. For example, that is the coronary artery diseases and atherosclerosis. Both of them are related to heart. But also there are pairs that are emerging through.
Let's wait a little bit more. It's coming.
Conexão de Atenas. É porque a gente consegue falar entre a gente bem, né?
É, eu acho que é para fora do Brasil. Ah, she's coming again.
Hello.
Great. Hello.
Ok.
So, and then we can identify a disease such as asthma and celiac disease that are not co-located, but they are interlinked by an immune response of IgA production. The transition of healthy to comorbidity state in an individual could happen because of various reasons, such as genetic or stochastic, or because of the environment. And it could also be driven by mechanisms such as loss of or weak protein-protein interaction and rewiring of network or emergence of disease model. So basically, we understand the disease comorbidity space in the context of aging is incomplete, as we really don't know much about the aging natural process and how this can interlink two or more different diseases. So to achieve this, we followed the strategy where we mine different data sets. So we integrated data from the Open Targets platform. So just to give a little introduction, Open Targets is a private academic partnership for target identification at the pre-competitive and preclinical stage. And this effort produces a platform that integrates a variety of different information to deliver associations between drugs and targets. And here we use from Open Targets, we use the disease and gene relationships that are basically the result of mining different types of data, from literature to protein structures and interactions to drugs and so on. We also use a single cell data set from the aging mouse that you may have heard of. It was published a year ago, and the tabula muris senis. That is the only really complete data set of the different tissues. It's a single cell data set of the different tissues and organs of the mouse across the different ages. I think it starts from the embryo up to the adult. And then using these data sets, we construct comorbidity clusters with their molecular mechanism based on this information. So we have so far applied these methods of developing these clusters to kidney. And first we identified the aging genes in kidney cell types. So from the tabula muricenis dataset, so we identified genes that are changing in expression across the different ages in the mouse, in the different cell types of kidney. And then we filtered, we used these to filter the disease gene associations from open targets. And therefore, we brought that down to about 551 age-dependent genes in kidney that are also associated with human diseases. And as a sanity check, we checked their biological process enrichment, and aging was definitely was picked up. And we also found that these genes were represented in the aging atlas, which is an externally curated database of aging genes. So that gave us some confidence to continue. So far, what we have identified is the aging-related genes in the different cell types in kidney, and we filter. So this picture here shows the different cell types, their aging genes, and then we successfully linked these to diseases. So these genes to the different diseases from open targets, and then we developed this disease network where the edges have a stronger weight according to the number of genes that are common between the different diseases. So basically, the steps here were disease to disease graph overlap count. And then we tried to identify different communities. We got a large network of diseases, and then we identified communities using Leiden method. So that is an example where this is the kind of network we derive, and then we further analyzed the communities with more than one member, and we started classifying the different diseases that we see associated with kidney age-related genes. Just to zoom in a little bit to show a section, a little community here. So in green circles, these are the diseases in kidney, and the prisms are the genes that link them to other diseases. And we can already see some links between kidney disease in kidney and also in other tissues as well. And for example, we find this APOL1 gene that is associated in kidney disease, and also the ACE that is also in kidney and aging. And also you probably have come across this, is also a target of coronavirus as well. And that is really where we are now. We're looking further into the different clusters and neighborhoods of diseases. And in the future, we plan to expand this to other tissues as well. and in order to generalize the principles of disease and comorbidity graphs across the different organs. This is it so far. So that's this gives a nice overview of what my team is doing. And finally, before taking any questions, I would like to thank my team, both the ones working on the Expression Atlas and also my research group as well, and especially our funders and our numerous collaborators in the different projects. So thank you very much. I'm happy to answer any questions.
Irene, congratulations for the work, excellent, and the excellent presentation as well. Well, it's amazing how many data you have and how many different things you are doing, sorry. Don't know if you have questions here in the chat. Well, students can ask in Portuguese and then we can translate to Irene if you wish. But before, let me see here. Yeah, and I don't see any question here, but I'd like to ask some questions to you.
Sure.
First one is about your expression atlas. Before the single cell expression atlas, that is very interesting, but you said we can query a gene in different tissues and conditions, etc. But you only told us about gene expression. Are you aware about isoforms, different isoforms of transcripts that can be expressed, differentially expressed in different tissues or conditions? Do you have any expectations in deconvolute the gene expression into different transcripts related to it?
Yeah, so that's something we have considered. That's a great question, Gloria. So we actually generate isoforms for the RNA-Seq experiments. So these can be downloaded from our FTP, but they're also available to visualize the expression of the different isoforms in the baseline experiments. So we don't, during our, well, we did some user surveys, and that seemed like a niche need for some of our users. So we do have it available within an experiment. So you won't be able to identify it by querying across all experiments. But within a baseline experiment, you can visualize the expression of the different isoforms. So now we haven't gone further to present them in the differential setting as well. But that's something I think we should consider for the future going forward.
Yeah, for sure. But for a single cell experiments is a little bit more difficult because the level of expression and the amount of reads you get from one sequencing result and one library, and then it's more difficult to analyze different isoforms in single cell experiments. But that will be great because I work with differential splicing, alternative splicing, and I would like to see this on your atlas. That will be great for us. We have questions here. Anderson asked if the data is available for everyone. I think you did some address, but go ahead.
Yes, the answer is yes. So all the data are freely available for everyone. That is the core of what we're doing. So, and there are different ways to access them. I think you can have a look at our FAQ. So there's a help pages in Expression Atlas, and if you have any further questions, you can send an email to the team for accessing the data.
Great, thank you. Guin Domingos has a question. I don't know if it's in Portuguese or in English. Would you like to show yourself? Guin.
Yeah.
Oh, hi, everyone. Good morning. Thank you, Irene, for your presentation. I'm from Chicago, Brazilian as well. And we are just starting this type of studies right here. So it was very clarifying, your lecture, I mean, your presentation, because I'm from the cardiovascular laboratory. So we are trying to start these new approaches for our experiments and our models, right? However, there is a lot of questions. One, the more important one for me, since I came from physiology, like basic physiology, and this is very, very, I mean, modern to us, if I can speak like that way. So this tool that you presented, we are able to use when we have our data and then make some comparison with the data available in the software and the program you showed us.
So you mean for single cell expression atlas or
Yeah,
the comorbidity?
Yes.
Okay. So if you generate, so basically, if you generate your own single cell expression data, what you can do is you can analyze it using the pipelines we provide. So you can either install them locally or you can upload your data to Galaxy and analyze them there. And we do provide tutorials as well and courses, online courses as well, if you wish to have a look. For Expression Atlas, it's more of a static tool. So we take publicly available data sets that they have been published in papers, and we just reanalyze them and load them in the database. So it's not really for interactive analysis, which is why we developed the instance in Galaxy to fill that gap, but it's separate to what Expression Atlas is trying to do.
Okay, I got you. Thank you very much.
No problem.
There is another question from Isabella. Isabella, would you mind to show yourself and ask your question to Irene? It's not necessary to show up, but I know that Isa always ask questions. Is Isa there? Well, I don't know. Maybe
No,
I can. Yeah, I can ask her a question. Isa said, "Thanks for." Yes, Ella. Sorry, she has no microphone. That's why she cannot ask. Isa said, "Thanks for the amazing presentation." And one question: why do you use Callisto in your workflow? Is it the best integration with Galaxy? Would you be using Salmon to produce the same results?
Right. So thank you for the excellent question. So why we use Callisto? So it's various different reasons why we select one tool over the other. So we do benchmark the tools and find out which one performs better. Obviously, something that doesn't produce accurate result wouldn't make it. We wouldn't use it. Salmon is a good tool, so it produces good results as well. So usually it's a combination of how much support the tool has by the community. So if we are sure that the tool is going to be continuously developed and maintained by the developers who produced it, then we might prefer it, or it might be it runs faster or requires less memory and so on. because we do run our tools on hundreds of data sets of different sizes and so on. So performance on the use of compute and so on really is important for us. So due to reasons like that, we preferred Callisto because it ticked all of the boxes. We didn't select it because it has a best integration with Galaxy. So basically, any tool we can integrate it with Galaxy. So I hope this answers your question.
Great. Any other question? Well,
Callisto,
I'd like
I think.
Okay,
I think that Callisto takes a little bit longer to run, but it's a little bit more accurate, right? So that's maybe because of that Salmon is more popular, because it's a little bit more rapid. Irene, can I ask a question, Gloria? Irene,
Sure.
Irene, I am very interested in a gene by gene querying. So when you query the single cell graph for a single gene, like you showed to us, I wonder. I see that you use some packages, but I wonder if it's possible to calculate a p-value or so that would relate the expression of the gene with the best way of clustering, because if I choose a gene, I have to change k equal 4, k equal 10, which one will be the best setup of the tool for us to have the most separated clustering of a single gene. Would it be possible to add a calculation for that to point to us, for this gene, use k equal 10?
So we don't really do that by gene. So it's really by experiment. So we so what we show by default, the k value that we show by default, is what we found to work for most experiments. And then we basically give a range of a few higher or lower k's, because that is actually quite a manual step in the analysis that we cannot really reproduce in a standardized way. Soon you will also have UMaps there. So we are, with the next release that should happen in a couple of weeks, there will be an option to show a UMap than a t-SNE plot, which can also help visualize the data better, perhaps. So yes, so that's what we are planning to do.
Because I think it's possible to calculate the best setup for clustering to separate the clusters for a single gene. But someone has to develop the parameters for that.
So we use the so ScanPy also recommends a k value, which we also use, but it's not per gene. It's for the whole data set. So I'm not aware, but please feel free to forward me any tools you're aware of, because that sounds interesting.
No, I think that someone has to develop that. We have a collaborator here that usually takes the clustering and analyzes which value of k-means, for instance, is the best to separate the cluster for that situation. So as you pre-compute all the data, probably would have this answer could be possible to have what's the best setup that will cluster nicely for this gene, present in this cluster, absent in this one, present in this, absent in this one. That's the suggestion for people interested in a single gene. in the single cell universe.
Great,
Okay.
thank you.
I have another question. There is no other question from the audience. It's about your network. That's so interesting, your disease network. And you showed about the aging in kidney and using different approaches. But you didn't comment that many other diseases associated are related to immune system. Do you think immune system is essential in aging, and then will be the basis for aging and other diseases?
So yes, you're right. So we do see immune pathways and the immune system coming up in all of these analyses, and it definitely plays a role, and it changes across aging. So that's something we haven't looked into detail yet on how this changes, but yeah, I think there is evidence in the literature already that it does change across aging, but how this relates to the particular diseases and the comorbidities is still unclear.
Yeah, and many pathways related to immune systems can be seen in neurodegenerative diseases like Parkinson, Alzheimer, and others. That's very interesting. And then I think we should take a look, more deep look into the immune system and pathways related to immune systems. That's great. Are you correlating this data with the literature that could be another layer in your network?
Yes, so actually the open targets data include a lot of literature annotations from text mining. So there is a layer within this, but we're also looking into aging databases. There are different databases that collect curated data from the literature of genes and aging diseases as well that we use as validation.
Okay, great. And there is another question, I think the last one, because we are going very further in the time, far in the time, from Patrick da Silva. Is there any ongoing project correlating the cell type and or tissue type expression level of each genes and the promoter sequence?
So I think, so I'm not aware of a particular project. But I think there are labs interested in looking at the cell types and the promoter binding sequences and how these affect the differentiation of the cell type and so on. I think there are also more single cell ATAC-seq datasets coming up as the technique has been developed further. So I think we will get more datasets of this nature.
Great.
You know, recently we had a talk from Ziping from Encode, and she has a nice database of regulatory elements. But the problem is, if you pick a gene, you can localize 3, 4, 5 enhancers around it. It's very difficult to match promoter, enhancer, and gene. You will have to deal with a network and then deal with a graph interpret that. But Ziping in ENCODE, they provide now a very great database for which enhancers are activating what gene. So you could download that and try to develop something, but be aware that you have some 4 candidates of enhancers regulating the gene that you want to look at. So it's kind of a difficult job to do that, but it's possible to explore. Yeah.
Mm-hmm.
Also,
maybe
we are talking with Ziping, and in ENCODE, they have a good source of data for transcription there that you do not have in the Atlas. I processed it, I downloaded the data from them and put it in your R data format, you know. So probably would be interesting to try to chase those informations for the Atlas. Mostly they have embryonic tissues for human tissues, not the adult ones. They have embryos, but lots of data there in the same format as your R data are. Could be a suggestion to try to include. It can send to you from where you download it. Yeah.
So we already include data from ENCODE. However, we don't have the latest version, so the latest release of ENCODE.
Yeah.
So every now and then we renew that. So that's probably up for.
Yeah, I use your data from ENCODE also, but not many tissues, right? Some 16 or so far. They have lots of data there. Could be nice to upload to the Atlas. I'll send you the info from where I got the information, the replicates and so on. Okay.
Okay, great. It was very, very nice to have you here. Almost 90 people in the audience. Many people are congratulating you, and it was very, very important for our graduate programs in bioinformatics in Brazil. Thank you so much for your talk. It was amazing. Thank you, Irene.
Thank you very much for the invitation, and thank you for the really nice discussion as well. It's been great to talk to you all.
Okay, hope
Thanks
to
a
see you
lot,
again
Irene.
soon. Bye-bye. Bye, people.
Bye-bye.
Have a nice day. Bye-bye.